Hello, everyone. So I just wanted to do a few small videos going over the exam objectives in preparation for our first exam. Now, to a certain extent, you guys have seen success in anatomy and physiology. That's how you got through Bio 201. So you know what it takes to prepare for these lecture exams. But then again, each instructor is a little bit different in terms of how they test and the format and that type of thing. Now we know that there's going to be 3 different sections to the lecture exam. It's going to be mostly multiple choice. It's going to be a short vocabulary section with words taken from our vocabulary list. And then it's going to be a short answer section where we have short answer questions taken from our exam objectives, and most notably the ones in bold. Those are our bank, if you will, of questions that you could possibly get as short answer questions. Okay. Now, I'm going to go through a few of these. I'm going to start with blood, and then I'll create separate videos for heart and for blood vessels and hemodynamics, just as a starting point for you, just so you can get an idea about kind of where to find these things and how the short answer questions work for this first exam. Okay. So in terms of blood, and I have a second monitor over here, so don't think I'm ignoring you, but I have a second monitor where it shows me the different bold objectives. And the first one says, distinguish between all the granular and agranular leukocytes. All right, so the deal here is that when we are talking about the blood, we know that we have cellular elements, and then we have the plasma, right? We have the fluid component, and each one is very important. And so what we're going to do is we're going to look through the PowerPoint, and we will find tables and charts that will be very critical for us in terms of bringing together the important information. And that's something that if you haven't noticed it as of yet, it is important. If the textbook publisher is going to take the time not only to write about the topic, but then develop some sort of a graphic, like we said, like a figure or a table that also details that topic, that means it's pretty important. And those graphs and figures and tables really bring the information together in one cohesive unit. So it's a nice way to really kind of coalesce that important content. So definitely pay attention to those. Okay, so the cellular elements. Now we start off with a slide like this one, which does a good basic job of introducing our cellular elements that we see over on the right, and then the constituents of the plasma that we see on the left. And that, the second bolded objective, actually asks you to describe the function of various molecules and ions found in the plasma. And so for that second one, that we'll kind of do a brief jump over to, this figure that we have here has a lot of basic information for you for the different ions and molecules that are important in the plasma itself. So on the left-hand side there, we see water certainly counts as one, right? And it acts as a solvent for carrying these other types of solutes around. We see the different ions, right? And some basic functions for those, even sodium, potassium. Those are some basic ones that you know about, that you may have heard of in terms of like the sodium potassium pump and things like that, and the intracellular and extracellular environment. When we get down to plasma proteins, remember there are 3 main plasma proteins: albumin, which helps to maintain osmotic balance, fibrinogen, which is part of the clotting process, right? So that we have the inactive form called fibrinogen that's soluble, and then ultimately that'll be activated to become fibrin. And then the immunoglobulins or antibodies that are part of our immune system or our bodily defenses. In addition, we have respiratory gases, hormones, nutrients, waste products, lots of different options for you to be able to list in a short answer question detailing what we find in the plasma and what's important. Now, I'll tell you that commonly our short answer questions will be about 5 points each. Not all the time, but commonly. And so that what that means is that there should be 5 detailed components to your answer, things that I can go through and go, yep, that's one, that's one, that's one, that's one. Was that 5? I don't know. But when you look at it, the short answer questions, you know, one of the reasons I put those in there is so that you guys can express in your own words what you've learned. But also, I try to create options in your short answer questions. And what I mean by that is For instance, we just looked at tons of different solutes that we find in the plasma, and a short answer question that's asking about it might only ask you to detail 5, right? And so you can choose any 5, whichever ones you remember, your favorite ones, whatever. So oftentimes our short answer questions will have that kind of flexibility built into them. And you want to answer as detailed as possible. Make sure you answer all parts of the question if it is a multi-part question. And, we hope that you don't get to this particular state. But if you don't have an answer for that short answer question, do not leave it blank, right? Always put in your best guess, because I'm going to be able to give you partial credit for whatever is correct in your answer. But to be sure, if you don't have anything there, then I can't give you any points because there's nothing for me to go on. Okay, so always make sure to put in an answer and have it be the best answer that answers the question itself. Okay, so we started out with this table, which gave us some information about cellular elements and those solutes that we find in the plasma. But we have more details about the cellular elements, which is what we were talking about, in future slides. And in fact, there's even a table, another table like this one, that does a good job of going through our cellular elements, our formed elements. So if you get a question that asks you to name and discuss 5 different formed or cellular elements of the blood, and we call them elements because they're not all cells. Remember that plasma are cell fragments, not whole cells. If you get that question and you're thinking, okay, wait, there's red blood cells, white blood cells, and platelets, what else is there? Do remember that of our white blood cells, our leukocytes, there are 5 specific types, each one with a different structure and function. So in truth, you have 7 different examples of cellular elements to choose from in order to answer that question. Right? So we have the red blood cells or the erythrocytes, which transport oxygen as their main function. As we talked about, they can transport things like carbon dioxide or hydrogen if they have a free binding pocket in their hemoglobin, but primarily transport oxygen. And then for our other ones, for our 5 other ones, our granulocytes that we see, these 3 here, as well as our agranulocytes, you can see the function listed right over here on the right-hand side. Same thing with our platelets. So flashcards are a really good way to just create study materials that you can review very quickly and get this information down. Name of the cell on the front, whether it's a granulocyte or agranulocyte, if we're talking about white blood cell, on the back, and then its function as well. And as you're creating these study materials, make sure that you're studying them in chunks, right? Don't do all 3 chapters of study materials all at once. Do it in smaller chunks so you can get that short-term repetition and you can really nail it down before you move to another topic, okay? Okay, so those are the first 2 in the blood chapter, the first 2 bolded or objectives that are in bold. The next one talks about anemia. Generally, we don't get into many diseases and disorders. We talk about the basic functions of the body and then only introduce diseases and disorders if they are particularly important or common or something like that. But when we get into anemia, there are a few different types, and those are listed right in our PowerPoint. I was trying to find them in the small windows over here. It's kind of tough to do because it's so small, but I found them. So you can see here in these slides that as a basic definition, as one of your vocabulary terms, anemia is a reduction in the oxygen carrying capacity of the blood. But there are various reasons that can come to be, right? So you see your different examples here: hemorrhagic, hemolytic, aplastic, iron deficiency, as well as pernicious anemia. So that is where you can get the answers to your question here in bold. The iron deficiency one is also sometimes called our nutritional anemia. So if you're not eating enough protein that contains iron, then that could affect your ability to make hemoglobin and then have functional red blood cells. Okay, so the last one in bold that we see for this chapter is asking about methods or mechanisms of coagulation. So you're kind of comparing and contrasting your 2 different pathways for coagulation. So in terms of this topic, I would say, on an aside, you need to know these first steps of what we call hemostasis, right? Maintaining the blood inside of you. Starts with vascular spasms for a larger blood vessel, platelet plug formation, and then coagulation. Those are the three basic steps where the blood vessel is trying to decrease the amount of blood that's being lost. But in terms of coagulation itself, remember that, for instance, when we look here, this graphic talks about the extrinsic and intrinsic pathways. And so you need to know the role of these different molecules that are listed in the exam objective. And so it says collagen, thromboplastin, coagulation factor, thrombin, and fibrinogen. And really, thromboplastin, you can probably even leave that one out for now. But the main thing we want to do, and this is common in our short answer questions, is you might be asked to do a compare and contrast. In these different ways, how are these two functions, these two processes similar or different, or how do they get this process done in these different ways? So we have an extrinsic and an intrinsic pathway. When we talk about the broken blood vessel, we need to be able to heal it on the inside of the blood vessel, as well as keep that blood that's leaking out into the tissues from in the surrounding tissues from continuing to flow out. And so the extrinsic pathways for the surrounding tissues, intrinsic is within the blood vessel itself. And so what starts these off, right? So what starts them off is in the intrinsic pathway, we have exposed collagen fibers because we've gotten to that outer layer of the blood vessel, which we shouldn't normally see, or even some chemicals associated with damaged endothelial lining of the cells of the endothelial lining in the inside of the blood vessel. Now, for the extrinsic pathway, it's where the blood is coming into contact with what's called tissue factor, which is found in the surrounding tissues. So from there, that's what starts them off. From there, we have two, if you will, kind of pathways that are similar, but a little bit different. So the intrinsic pathway is actually a little bit slower compared to the extrinsic pathway. There's a whole bunch of clotting factors, right, that some of them are listed here. You hear of factor 10 and 13 and 2 and all this type of thing. You don't need to know all those. But what you do need to know is that the intrinsic pathway uses all of the factors, the complete sequence, whereas the extrinsic pathway uses a shortened set of those factors so that we can kind of sandbag off, like if you think about flooding, sandbag off that surrounding area. So the extrinsic pathway is faster because it uses fewer factors, whereas the intrinsic pathway, is a little bit longer because it uses the full pathway and all the factors. But ultimately, like you see in this figure here, the ultimate result is that we get the activation of this enzyme called prothrombinase, and prothrombinase as an enzyme activates thrombin, and thrombin activates fibrinogen. That plasma protein that we talked about earlier called fibrinogen now it starts out soluble, now it comes out of solution and forms these protein threads, fibrin, that reinforce the platelet plug that is already formed to form a strengthened clot. Okay, so both of these pathways lead to the activation of fibrin from fibrinogen. Okay, so in any case, those are the first bold exam objectives and potential short answer questions for the blood. So I'll end this video here, and then what we'll do is we'll go and create a couple more for heart as well as the blood vessel chapter.